based on the nature of microbiome data. So I just formed these key analytical domains. And I was also mindful not to have too much crossover with the Aristotle. And obviously, when we do the integration, the multi-omic analysis, we want to make sure that we're not having a lot of overlap. 13 key analytical domains are the overall microbiome. We'll go through each one of them. Core microbiome, community diversity, resilience, the archaeome. So we're breaking it out by kingdom. The microbiome, fungus, obviously, the virome, and then we get into some more standardized panels like the probiotic panel at the strain level, and then we'll go through the pathogen and parasite panel, which I think is very, very detailed compared to other players in the market, including GI maps, based on the importance of the gut microbiome and things like digestion and nutrient generation. I thought those were an obvious inclusion. Gut barrier integrity, obviously, and that dovetails nicely into our inflammatory panel, pathways, and including some visuals. So I do have a conceptual summary, bringing together all of the analytical domains and putting it into a figure, basically. The digital twinning personalized protocols are in tabular format. They're not as we currently have them in the Aristotle. So there are going to be some separate degrees of separation here with how these different components of the report are presented. The appendix, again, because of how complex the microbiome is, a static fixture of, I right now have the top 30 gut microbes that are detected in a sample. Reference population, I'm using data from the Human Microbiome Project, very well validated, very robust, gold standard. Basically just looking at population norms and kind of what that means in the context of the person's health. And then testing methodology and references. Moving from here, we go to basically the main dashboard showing the overall score of the overall score, and then the score broke out by the 13 analytical domains. This as a separate page where we put the key recommendation for each domain. You can print it out. It's just a really quick reference. I think this format makes a lot of sense, and I'm a bigger fan of it because I'm going to probably be a little bit more verbose with my recommendations, and we're going to have a big problem fitting all that content into one page. From here, so there's also an educational component that is really important with the microbiome, any omic layer that we do, and I'm trying to fold that in to the report, but again, not being too verbose. So here we talk about how your overall landscape looks. We go over the number of species that we detected in your sample, breaking it out by kingdom, really showing what the configuration of your microbiome looks like at the kingdom level. And this segues nicely into the analytical domains where we break these out further. So from this, someone can start educating themselves, understanding, okay, I understand the microbiome is comprised of microorganisms of these different kingdoms, but really, who are the main players, right? And obviously, it's going to be fairly individual, but bacteria reign supreme. The sample, we're not going to find anyone that has, less than 90% of their relative abundance from bacteria. So I thought this was nice. From here, we have the core microbiome. So this is really looking at the bacteriome, and I might change that because, again, that just based on the total abundance. So we're going over what we detected in the individual sample. So this will be specific to the person, what that taxa is, and what its contribution is to the overall community. We are bringing in some ecological metrics, and there is a little bit of explanations here for richness, captured via alpha diversity, and dissimilarity captured via beta diversity. These are benchmarked against the Human Microbiome Project, so you can see where someone's falling in that range. Our resilience score, so informing this is microbial diversity, functional stability. So we are able to annotate functions with our shotgun metagenomic sequencing. Functional redundancy, pathogen resistance and control, microbial interaction networks, which I do have network figures in the report, which I think, again, is another difference or departure from other tests out in the market. And then obviously the potential of metabolite production, because we're not currently doing metabolomics. Oh, and another thing, you'll notice I don't put any time recommendations for the individual domains. We're going to have one recommendation provided in the digital twinning section of the report. So that was another piece of feedback that we got. Okay, so for the archaeome, again, we're doing a little bit of education. What this is, the paragraph at the bottom is going to be describing what we found in the person sample. So we're going to be taking the functional data and then the taxonomic data from the archaeome and doing a network analysis. Actually, what I'm using now is MMVec, which we've published on that before. So this is a neural network based analysis. So we can get a really good handle on what's going on with the archaeome and how we might want to modulate it when it comes to the digital twinning section. Same thing for the microbiome. We know that fungus, of course, is really emerging as a very important component of this environment. And no one, to my knowledge, is breaking it out like we're doing in this test. I mean, they might have like species or even in some cases strain levels listed, but they're not really looking at the overall picture of these different kingdoms. Virome, again, another really important piece, particularly when we start talking about bacteriophages, which we do annotate. You'll see like with what we're doing in the probiotic panel, because some of those can be probiotic. And then we have the probiotic panel. We have bacterial, we have fungal, and we go into the pathogen and parasite panel. So again, I think this is pretty robust. We break it out by kingdom. So we have our bacterial, our fungal, viral pathogens, protists. So we are able to get pretty granular here. And then our parasites, everything that we've heard from people that want this type of panel, from roundworm to pinworm to tapeworm to whipworm, any kind of worm you want, we have here. In terms of actually detecting these, this is very robust because the DNA content it's either there or it's not. So I think this is a really good approach. Here we have digestion score, similar to what we had previously. We have these sliding bar charts. We have all the important functionalities such as fiber degradation, protein fermentation, gas production, lactose and simple sugar digestion, fat metabolism, bile acid metabolism, enzymatic function, and mucus degradation. From here, I know it's a little similar, but I think it's important to break these out. So nutrient generation, we know that the microbiome not only contributes in terms of fuel substrate via short chain fatty acids, as I have here, the main constitutional ones, but also modulations of amino acids and some important vitamins such as the different B vitamins and of course vitamin K. And then we have the gut barrier integrity. So all of the main components, the subcomponents that would inform this score, butyrate production. And there's going to be some crossover, obviously, with some of these scores just inherent to the metric that we're looking at. Mucus degrading bacteria, pro-inflammatory, anti-inflammatory microbes, leaf polysaccharide producing bacteria, tight junction regulation. Again, these are inferred. And same thing with the microbial metabolites. And then the last is the inflammatory panel. Some unique components here, such as hydrogen sulfate production, tryptophan metabolism, inferring cytokine production. I think this is a good encapsulation. Okay, so the conceptual model, this is fairly simple. Again, kind of getting people's minds are like, yeah, you've read all this text, you've seen the sliding bar charts, you've seen the networks, but just to see this visual, I think is going to be pretty powerful because in the lumen, where the microbial contents reside, we can put call out boxes saying the constitutional bacteria, whether they're pathogenic or commensal, whatever. We can vary the level of mucus that we infer based on the sample. We can talk about the integrity of the brush border, and then the important signaling agents that are potentially coming into circulation and interacting with host tissue.
Yeah, it's all there.
So
and all the components are in this.
Yeah, so this is the conceptual summary, bringing together all 13 key analytical domains. From here, the digital twinning section, we're not going to break it out by domain because some of these recommendations are going to capture several domains. So we'll have the recommendation and then the domains that that recommendation falls within, and then we'll have an important score in terms of what you should be. Like a hierarchy, I was going to actually do almost like a cluster with awards, but if we're going to provide an important score. And the length of this will vary So right now, as a placeholder, there's 2 pages devoted to this. Okay, so for the appendix of the keystone microbes, I think I just want to do sliding bar charts. I have 3 pages of this. I'm thinking 30. I think for right now, this is probably the way to go. But what does deficiency mean, though? I mean, not every, you're assuming that everyone should have a global average microbiome. That's not the case at all, you know. And I think that's the kind of the wrong way to approach the microbiome. Functionality is really what you want to hang your hat on.
Yeah, especially the microbiome. Yeah.
We almost kind of have to break the paradigm of thinking, because you can't approach this like a normal clinical test. We can say what's normal for functionality. We can't say what's normal for the actual microbes themselves, because you can have vastly different microbiome populations in 2 different individuals, but have a very similar functional capacity. But in terms of like one healthy microbiome profile, at the taxonomic level, that doesn't exist.